In our class today, we are going to look at acidic hydrolysis of glycogen and iodine starch test. We start our introduction by looking at the structure of glycogen. And from the structure, we can see that glycogen is a polymer of glucose residues linked by alpha 1,4 glycosidic bonds mainly. So these are the alpha 1,4 glycosidic bonds over here, and alpha 1,6 glycosidic bonds at the branch point. And this is the branch point of the glycogen structure. Also, what we have to note is that glucose is stored as glycogen predominantly in liver and muscle cells. And about 70% of the total glycogen in the body is stored in the muscle cells. Acid hydrolysis of glycogen. First of all, what we have to note is that acidic hydrolysis is simply the reverse of esterification. And it involves the heating of ester bonds with water containing a strong acid catalyst. Also, complete acid hydrolysis of glycogen will convert glycogen from a non-reducing substance into reducing substance. So now let us look at an example. The complete hydrolysis of glycogen molecule with acid results in the splitting of all its glycosidic bonds, giving only glucose molecules as the product, and the glucose molecules happens to be the reducing substances. Also, apart from acidic hydrolysis, glycogen can be broken down into D-glucose subunits by enzyme catalysis. And these enzymes are more specific in the type of bond they split. Example, in animals, the enzyme phosphorylase catalyzes the breakdown of glycogen to phosphate esters of glucose by cleaving to the alpha 1,4 glycosidic bonds. Acid hydrolysis. Acid hydrolysis is a hydrolysis process in which a protic acid is used to catalyze the cleavage of a chemical bond through a nucleophilic substitution reaction in the presence of water. So, for example, in the conversion of cellulose or starch or glycogen to glucose. And what you also have to note is that protic acid is that acid which donates proton, or in other words, an acid that releases hydrogen ion, also called ozonium ions. Now, so we have for the protic acids, we have three main types. So we have the monoprotic acids, and these are acids that donates one hydrogen ions, and these are the examples over here. We also have the diprotic acid, and they donate two hydrogen ions, and we also have the triprotic acid, and they donate three hydrogen ions. Glycogenolysis. So when we talk of glycogenolysis, is a process by which glycogen is broken down into glucose. And what we also have to note is that glycogenolysis helps to provide energy to the body and helps to maintain the body blood glucose level. So as you can see from this reaction over here, or from this picture, we can see that the enzyme alpha 1 6 glucosidase in the presence of water catalyzes the breakdown of the glycogen to a glucose unit and a glycogen unit, which is shortened by one glucose residue as seen here. Also, in the enzymatic degradation of glycogen, the enzyme glycogen phosphorylase catalyzes the breakdown of glycogen to glucose 1 phosphate, which is a phosphate ester of glucose, by cleaving to the alpha 1,4 glycosidic bond of the glycogen molecule. So, as you can see here, this is our alpha 1,4 glycosidic bond of the glycogen here, which the enzyme, the glycogen phosphorylase, will cleave to catalyze the breakdown of this glycogen to produce glucose 1 phosphate, or the phosphate ester of glucose, and a glycogen unit, which is shortened by one glucose residue as seen here. Iodine test for starch and glycogen. So first of all, what we have to notice is that iodine solution or reagent can also be called as potassium triiodide solution or potassium triiodide reagent. So first of all, what we have to notice is that iodine, which is a small nonpolar molecule on its own, is insoluble in water. Therefore, potassium triiodide solution, which is iodine dissolved in potassium iodide solution, it's used as a reagent in the starch or glycogen iodine test. So let's look at a detailed note on the iodine test for starch and glycogen. So first of all, what we have to note is that the use of iodine reagent or solution is useful to distinguish starch, dextrin, and glycogen from other polysaccharides. So iodine reagent yields a blue-black color in the presence of starch, a reddish purple in the presence of dextrin, and reddish brown color in the presence of glycogen. And what you also have to notice is that other polysaccharides, apart from these mentioned, and monosaccharide, yield no color change. The test solution remains the characteristic brown-yellow color of the reagent. Also, starch and glycogen are able to form helical coils, and these helical coils formed allows iodine atoms to fit into the helices of the starch or glycogen to form a starch iodine complex or a glycogen iodine complex. What you also have to notice is that starch in the form of amylose or amylopectin has less branches than glycogen. So this means that the helices of starch are longer than that of the glycogen, therefore allows more binding of the iodine atoms to it than the glycogen. And the result is that the color produced by a starch iodine complex is more intense than that obtained with a glycogen iodine complex. So now let us look at the test protocol. So we'll start by looking at the materials needed for the test protocol. So we have 2 molar HCl, we have 2 molar sodium hydroxide, we have glycogen of this concentration, we have our iodine solution, we have our pasture pipette, We also have our white towels and we have our boiling water bath as seen here. So let us start by looking at the methods. First, pipette 1 ml of glycogen into a series of 7 test tubes and label them. To add 1 ml of 2 molar HCl into each tube and place in a boiling water bath of say 80 to 100 degrees Celsius. 3. Remove a tube at intervals of 0 minutes, 5 minutes, 10 minutes, 20 minutes, 40 minutes, 50 minutes, and 60 minutes. 4. Neutralize the contents by adding 1 ml of 2 molar sodium hydroxide. 5. Test the presence of reducing sugars in each tube using the starch or glycogen iodine test method. So, so let's see how to perform the starch glycogen iodine test. So first is to withdraw a drop of the hydrolyzed samples and spot onto a drop of iodine solution on a white towel. The next is to repeat the procedure for the other tubes. And finally, observe color intensity and comment on the extent of the hydrolysis. So this is just a picture showing how to perform the starch glycogen iodine test. So let us look at some keynotes in the test protocol. So first of all, what you have to notice is that adding 1 ml of 2 molar HCl into each tube causes acidic hydrolysis, and heating in a boiling water bath aids or speeds up the hydrolysis. 2. The acidic hydrolysis of glycogen using HCl and heating increases with respect to time, thus from the 0 minute to the 60 minutes. And this, as a result, decreases the color intensity during the glycogen iodine test, thus increasing incubation time, increases the amount of glucose produced, hence less glycogen available. And this results in a decrease of the color intensity with respect to the time of incubation from the 0 minutes to the 60 minutes. Now, so this is just an example of how your test result will look like. So over here, I am using 0 minutes to 22 minutes, but you have to keep in mind that from the test method, we are using from 0 minutes to 60 minutes. So as you can see over here, from 0 minutes to 2 minutes to 4 minutes to 6 minutes to the 22 minutes, there is a decrease in the color intensity as seen over here. Thank you.